So this is your regulatory gene, it has its own promoter. This is your regulatory protein. It binds on the promoter, basically it binds on the operator region. So operator is nothing but a part of the promoter. So if you have a promoter, you know transcription factors, RNA polymerase, all of them, they bind there. So one region of the promoter is referred to as an operator, and obviously if the regulatory protein is binding on the operator, there should be some sequences that it recognizes. And then once it binds, it helps in the transcription of the genes called the structural genes. Here in this case, when we looked at lactoferrin, we said these structural genes are synthesized, and once the structural genes are synthesized, they help in metabolizing lactose. And that's why it's called lactoferrin. So the regulatory protein can bind here on the operator region, and it can either promote gene expression or it can repress gene expression, right? This is what it can do. Now the structural genes are the ones that are involved in metabolism. And once the regulator binds, all these genes, gene A, B, and C, are all synthesized as one single messenger RNA. So you have, from this one single messenger RNA, you have 3 different proteins. And if you're considering this as a case of lactoferrin, these 3 proteins will actually metabolize lactose. So that is about gene regulation in bacteria. So this is the regulatory protein. Now, so sometimes these regulatory proteins would need something called an inducer, which this inducer will actually bind this regulatory protein and prevent it from binding the operator. So here, this is your regulatory protein once again. It normally binds on the operator, and it can, it can actually prevent transcription from happening. So look at this here. So this is the regulatory protein. It binds here, and there is no transcription. In such a case, sometimes this regulatory protein can have a positive effect, or it can promote transcription, but in this case it needs this inducer. The inducer will bind the regulatory protein and actually prevent it from binding the operator so that transcription can take place. So the regulatory protein can activate transcription, but it can also repress transcription. Depends on what the role of the regulatory protein is there. Depends on what the role of the regulatory protein is for that structural gene. So the binding of the inducer here actually prevents the operator from binding. So if you consider lactose as an inducer, it actually works in the case of lactoferrin. So if this inducer here is lactose, it binds the regulatory protein and prevents the regulatory protein from binding the operator, so you have transcription. So this is an inducer. In some cases, instead of an inducer, we have something called a co-repressor. Here in this case, there are some other proteins or some metabolites which can bind to the regulatory protein. Only then it can bind the operator. In the previous case, if you have an inducer binding the regulatory protein, it cannot bind the operator region. But here, if this co-repressor is required actually to bind the regulatory protein, thereby it can actually bind the operator region. So we looked at regulatory proteins, structural proteins, regulatory proteins, structural proteins, inducer, co-repressor. These are the 4 things that I spoke for chapter 16, right? I'm not going to go through all that, but we know that's what this is all about. Now here, the reason why I went through this 5-minute quick review is because all chapter 16 is regulation in prokaryotes, like bacteria. Now, chapter 17 is regulation of gene expression in eukaryotes like us. Okay? What really regulates gene expression? In fact, most of chapter 17 is what you have covered in the previous chapters here and there. So what you're going to hear in chapter 17 with gene expression in eukaryotes is not something very new to you. So you'll be looking at, what really regulates gene expression. Obviously, I'll be saying about transcription factors, which actually regulate gene expression in eukaryotes. I'll be speaking about histone proteins. I'll say that acetylation, methylation, methylation represses transcription in eukaryotes. We'll speak about acetylation. We'll also speak about methylation, not just to histone proteins, but methylation can directly happen to DNA itself. So we're going to speak on that, and we're going to see the role of how some RNAs like microRNAs and small nuclear RNAs, and all these RNAs can also regulate transcription. So, like I said, all these concepts which I have talked to you in just a minute is something that we have covered in the previous chapter. So we will be looking at those concepts, and we'll be now saying that these are the factors that lead to gene expression in eukaryotes. Now, so with gene expression in eukaryotes, many bacterial and archaeal genes, they use operons, because a bunch of genes can be one common promoter, regulator binds, gene expression happens. So that's what we refer to as operons in eukaryotes. Now, sorry, prokaryotes. Now, chromatin structure, when you use the word chromatin, chromatin actually is something that is synonymous to eukaryotes. So chromatin is nothing but condensed DNA, right? Condensed form of DNA is usually seen in eukaryotes. So chromatin structure affects gene expression. And also the presence of a nuclear membrane also regulates gene expression, because once you have RNA synthesized, it undergoes certain modifications inside the nucleus, and then it is actually transported into the cytoplasm. Whereas if you look at prokaryotes, you don't have a nuclear membrane or nuclear envelope, and there is not so much of special processing that happens to the RNA before protein synthesis happens. But here in eukaryotes, inside the nucleus, before the RNA moves out, polyadenylation happens. At the 5' end, you have a 5' cap, which is nothing but methylation of the DNA. Then you have some other special nucleotides added, and then it's moved out into the cytoplasm, eventually for protein synthesis. So nuclear membrane also plays a role, because the nuclear membrane differentiates the cytoplasm from the nucleus in eukaryotes. But whereas prokaryotes don't have a nuclear membrane. Now, also DNA's 1 hypersensitivity is actually a measure to say if a gene is undergoing, if a gene expression is happening or not. So what do you mean by DNA's 1 hypersensitivity? DNA's 1, as the name implies, is actually an enzyme that digests DNA. It can only digest DNA if DNA is relaxed, so to speak. So let's say you have DNA coiled around histone proteins very tightly. The DNA's 1 enzyme cannot sit on the DNA because it's coiled around the histone proteins. But let's say if a gene expression is happening, basically let's say protein synthesis, RNA synthesis is happening, then obviously the DNA should uncoil from the histone proteins and should open up. When DNA opens up, DNA's 1 enzyme can actually act on it. So let's say if you were to add DNA's 1 enzyme, and if DNA is digested, you can in a way say that gene is active. Basically gene expression is happening because the DNA has opened up, and that's why DNA's 1 enzyme is able to act on it. So adding DNA's 1 and ensuring that DNA is digested is a way to say that gene is actually undergoing active transcription. So this relaxation of chromatin structure allows regulatory proteins to act and bind DNA sites. At least 3 different processes affect gene expression. One is chromatin remodeling, which is obviously addition of histone proteins or removal of histone proteins. Chromatin remodeling is nothing but coiled and uncoiled. Modification of histone proteins. So when you speak about modification of histone proteins, you're referring to the addition of methyl groups or the addition or removal of acetyl groups. Methyl groups and acetyl groups are categorized under histone protein modification. And then there is a third classification called DNA methylation. Remember, DNA methylation is different from histone methylation because histone proteins, if it's a protein at the tail of the protein, basically the last few amino acids of the protein, you sometimes find a lysine amino acid to which methyl groups are added. That is called histone methylation. But here when you say DNA methylation, the cell machinery actually directly adds a methyl group to the DNA itself. When you have methyl group added to histones or to DNA, it usually represents a repression. RNA synthesis is not happening. Methyl groups prevent transcription most of the time. Most of the time, almost, because there are a few exceptions where addition of methyl group to histone actually promotes transcription, but usually methyl groups actually repress transcription. Now, looking at these three, what is chromatin remodeling, histone proteins, and DNA methylation? First one is chromatin remodeling. So, some transcription factors and some other, let's say, regulatory proteins, they alter the chromatin structure without altering the chemical structure of the histones directly. Because some transcription factors, some proteins actually have the ability to kind of penetrate into histone bound DNA and bind the DNA in such a way that they open it partly, allowing other transcription factors and RNA polymerase to sit on the DNA and start reading it. So here, these transcription factors are not removing the methyl groups on the histone protein and not opening up the DNA. They're directly penetrating into the DNA, to the complex DNA, and they move the histone proteins aside, thereby giving way or making place for RNA polymerase to bind and start reading the gene. They bind on DNA and reposition the nucleosomes. Basically, the transcription factors is pushing the histone protein complex so that RNA polymerases can now bind and start transcription. Okay, that is called chromatin remodeling, where transcription factors can penetrate. And here we are not speaking anything about histone modifications. Look at this here. See, this is tightly bound DNA to histone proteins, which are shown in pink. Now, chromatin remodeling complexes. Some transcription factors shown here in brown, this circle here, using some energy in the form of ATP, they kind of bind on the DNA in the middle, thereby they push these histone proteins aside. Now they have opened up the DNA in such a way that some other transcription factors and RNA polymerases can bind here and read the gene. So that is done by some of these chromatin remodeling complexes. And when you say chromatin remodeling complexes, you are once again referring to some other transcription factors. It's all about one or other transcription factors which are actually binding to the DNA directly without changes to any of the to the histone proteins. They enable transcription to happen. So look at this. So once this chromatin remodeling complex, which is a transcription factor, binds the DNA, it pushes these histone proteins, makes way for this RNA polymerase to bind on its consensus sequences, and thereby you have messenger RNA synthesis, which is shown here in green. Now, the second one is histone modifications. So, histone, is an octamer. Basically, you have 8 histones. Basically, you have 4 histones in duplex form. Basically, you have 4 histones with 2 pieces of each, which is an octamer. And to this 8 units of histone protein, DNA binds around it. The histone in the octamer core of a nucleosome have 2 domains. One is called the globular domain, that is associated with other histones and DNA. And the second is actually a tail domain to which different groups can be added. Okay, the globular domain of a histone protein associates itself with other histone proteins and also kind of links itself to the DNA. Whereas the tail domain of a histone protein is where you can add a phosphate group, a methyl group, or acetyl group. I mean, phosphate groups are not so commonly added, but usually it's methyl and acetyl groups. Okay? Now, when you add a methyl group, which is represented here as CH3, usually it promotes repression. Most of the cases. And this is because of an enzyme called histone methyl transferases. What these methyl transferase enzymes do is that they transport a methyl group and they add it to the lysine amino acid on the tail of the histone protein. Once a lysine amino acid, once a methyl group is added to the lysine amino acid, automatically the histone proteins, they begin to coil around DNA and tighten the DNA. Thereby they ensure that DNA is not accessible. So now transcription is repressed. So that is by the addition of a methyl group, which is done by methyl transferases. So acetylation, which is CH3CO, this group, addition of an acetyl group actually promotes transcription or stimulates transcription because it destabilizes the chromatin structure. In other words, destabilizing is nothing but this is DNA tightly bound to chromatin. And when you see this AA all here, all these acetyl groups are added, you see the DNA in between is actually exposed very easily because some of the histone proteins have been pushed away. Or they unwind themselves from the DNA and move away. When you have an acetyl group added to the tails of this histone proteins, the histone proteins unwind from the DNA, thereby making it accessible for transcription. So all these are actually different levels of gene expression in eukaryotes. I always say that here because, like I said, the previous chapter is about prokaryotes, and we spoke about OCON, structural genes, operator, and all these. Now here we're looking at gene expression in eukaryotes. One example of as to how acetylation plays a role in gene expression is shown here in the flowering plant Arabidopsis thalania. This Arabidopsis is a plant which is very well studied for developmental biology as to how gene expression happens during development. So also for development, people study mouse, people study Drosophila for flying models or animal models, but for plant models for development, people, a lot of scientists have studied this plant called Arabidopsis thalania. Here, these two slides are actually meant to quote an example as to how acetylation regulates gene expression. Here, they speak of a gene called Arabidopsis flowering locus C. So that is actually a gene that codes for a repressor protein. So basically, if this gene codes for a repressor protein, what does that gene do? It prevents activity of some other genes. So now, this Arabidopsis flowering locus C, which is FLC gene, which if it is a repressor, it prevents the plants from having flowers. So if this gene is expressed, since it is a repressor, it prevents the plant from having flowers. Now there's another gene called flowering locus D. What it does is, it represses the FLC, which means flowering locus D, which is the second gene, it represses the repressor. You get that? It represses the repressor. So basically, if this is a repressor gene, now this flowering locus D, it represses the repressor. Now how does it do it? This flowering locus D gene, it codes for an enzyme which is deacetylase, which basically deacetylases, they remove the acetyl group. I said the presence of an acetyl group actually promotes transcription. Basically, gene synthesis happens, a protein synthesis happens with the help of acetyl groups. Now, how does this flowering locus D, which codes for a deacetylase, prevent the activity of this FLC? What this deacetylase protein does is, it removes the acetyl groups from the FLC. Once you remove the methyl groups on FLC, since the FLC gene has lost the acyl groups, automatically histone proteins will bind to this gene and will prevent the gene from expressing. So now you have a situation where the repressor, this repressor gene is repressed because it lost methyl group, and that's because of this deacetylase enzyme which is synthesized from flowering locus D gene. So now since FLC has lost its methyl groups, that protein is not expressed anymore. So since it's a repressor, now in the lack of this FLC, the plant begins to have flowers. Is that clear? Yes, is that clear?
Yes.
Okay. Okay, so look at this. FLC gene. It codes for a transcriptional repressor protein. If you look at this plant, there are no flowers. Now FLD, it codes for a protein which is a deacetylase enzyme which removes the acyl groups. So this blue protein shown here, which is an enzyme, it acts on the FLC gene, right? It acts on the FLC gene and removes the acyl groups. So automatically, this repressor protein is repressed, and now you see this plant has flowers. They're kind of whitish, you don't really see them very clearly here. But yes, so the plant has flowers because the repressor is repressed. Now this is an example for the previous two slides. So we spoke here about DNA methylation, we spoke about DNA acetylation, and to quote for an example of how acetylation or deacetylation can regulate gene expression, we looked into the flowering plant Arabidopsis thaliana. Okay? Now, the other way you can actually study about gene expression, I mean, how do you know that a certain, let's say a certain transcription factor is binding the promoter of a gene? I said that transcription factors bind the promoter, they enable the RNA polymerase also to sit on the, to recognize the consensus sequences and read the gene. I said transcription factors do that. But how do you know that transcription factors are enabling gene expression? For that, you do an experiment called chromatin immunoprecipitation assay. This assay, this experiment actually helps you to understand how, it tells you how, if a transcription factor is sitting on the promoter of a gene or not. So, now, transcription factors and other proteins bind the promoters and enhancers. So, for chip assay, what you do is, it actually tells you if the protein, here when I color the protein in green, it's referring to a transcription factor here. If this transcription factor is binding to the DNA. For example, in my work when I did for my PhD work, I did this chip assays. So my work was, I think I keep saying PAC6 transcription factor. Did I say that several times earlier? Yes, PAC6. I worked on PAC6, which is a transcription factor, and it regulates the expression of many genes. One of my projects was to see if PAC6 regulates the expression of a gene called CRALBP, C-R-A-L-B-P, which is cellular retinal head binding protein. It is expressed in the eye. I was studying the role of PAC6 in the brain. So I wanted to see if PAC6 binds the promoter of CRALBP in the brain. For this, what I did was I took embryonic day, basically pregnant mice were actually sacrificed and the fetus was removed. I did that. So you take the brain of this fetal mice, and yes, I know it's bad, but then that's a part of my work. I mean, yes. Yeah, I was about to say something, and it doesn't matter now. It's not so nice to hear. But okay, you hold the mice by the tail. You put something on its head and you pull it.
Oh.
I did it so many times. I really hated that part. So you kind of.
I'm gonna have nightmares.
Sorry?
I'm gonna have nightmares.
Sorry, but yeah, I didn't like it at all, but I had to do it, and I had an ugly face when I did it.
That's normal.
I mean, science has to advance. If not for that, we wouldn't have aspirin for your headache. Let's put it that way. We wouldn't have drugs for cancer. We wouldn't have. I mean, someone has to make a sacrifice. Fortunately, it's not you and me. Some other vertebrates like plants. Okay. So from the fetus, what he did was you take out the brain tissue of this fetus, and at the stage where you see PAX6 is very active. I knew that at embryonic day. Okay, this part of the slide is not something that you have to memorize for the exam. I'm just giving you the steps, so just listen to it like a story. So at embryonic day 14 or 15, I mean, you represent it as 14.5 and 15.5, not 14 or 15 exactly. That's how they do it in the study. So at that embryonic stage, when the mouse are not born yet, PAX6 is very active. So I take the brain tissue at that stage and add something called formaldehyde, which is, if you go to the zoology labs, you see the snakes dead and you have it in this glass chamber standing like that. So all these dead animals which are stiff in place, it's because of formaldehyde. If you add formaldehyde, tissue is not degraded. So I used to add some amount of formaldehyde to this to the brain extract, and automatically what formaldehyde does is, if a transcription factor, if any protein is sitting on the DNA, or if any other protein is linked to any other protein, or protein linked to DNA, or anything is linked to anything, it kind of holds it in place like that. It kind of makes it like a cardboard. So if you take a mice and if you pass a lot of formaldehyde through the mice, you kill the mice, and you can actually take the tail and kind of lift it up like that. It's going to stand like a flag. So basically, in other words, formaldehyde makes tissue like a cardboard. So when I added formaldehyde, if transcription factors are sitting on the DNA, something like that, it's going to freeze in place, so to speak. Then I adds protease inhibitors, which, let's skip that step, it's too long. So you add PAX6 antibody. So this antibody binds the PAX6 transcription factor, and I precipitate the whole thing through centrifugation, and then I add a lot of salt. When you add a lot of salt and incubate at a certain temperature, what happens is these transcription factors, which are strongly bound on the gene, they kind of are removed away. So now you have a piece of DNA that's precipitated now because I got rid of the transcription factor. Now, if this is the promoter of CRELBP gene or any other gene, you can do a PCR with very specific primers for that region. If you have a band on a gel, you know that you have precipitated the CRELBP promoter region. Because you know the promoter region of CRELBP. For example, if you look at it, I'll come to this slide in a second. So if you look at this, see, this is exon 1 of CRELBP gene, my work, okay? And upstream of the promoter, you see this spot here, these two spots here, this one and this one and this one. These are consensus sequences, which I marked with that, black rectangle spot, whatever that is. And then on top, you see these arrows. These arrows are primers. So I did the formaldehyde addition, then I add the PAX6 antibody, I slowly spun it, pulled everything down, then I used salt, got rid of the transcription factor, used the primers specific for CRELBP promoter. So when I did 5 different PCRs with 5 different promoters, you see I had very strong bands here and here, which indicated that piece of the DNA was precipitated, indicating that your PAX6 transcription factor was actually sitting there. So doing this experiment called chromatin tubular precipitation assay, you can actually find out if transcription factors are sitting on the promoter of a gene or not. Is that simple? Now here's the experiment. So DNA binds protein, which is transcription factors binding protein. You cross-link, in this case with formaldehyde, and then you break the DNA into pieces. I skipped that step, but yeah, you break the DNA into pieces by something called sonication using very high frequency of sound. If you expose a cell sample to very high frequency of sound, that vibration actually breaks the DNA into pieces. Now we have pieces of DNA with transcription factor bound on it. Now to know if it's bound on it, I use this one in red here, which is the antibody. This antibody binds this transcription factor here, and then when this whole complex can be precipitated by very gentle centrifugation. Now you get rid of these transcription factors and the PAX6 antibody with salts. And then here, this piece actually they sent for sequencing. In my case, instead of sequencing, I did a PCR with specific primers, and then that's how you know if transcription factors are binding the promoter of a gene. So chip assays is a technique which helps to determine the location within the genome where a specific protein interacts with DNA. So this is a way for me to know that PAX6 is regulating the expression of a gene. Let's say transcription factors are expressing the regulator of a specific gene. Okay? Now, There are other things that also regulate gene expression. The other thing is DNA methylation. The previous one was an example. Previously we looked at histone methylation, right? Addition of methyl groups to histone. Here we're going to speak about methylation of the DNA directly. So if you add a methyl, if a methyl group is linked to the DNA directly, to let's say to the nucleotides, in this case also the gene expression is repressed. So DNA methylation is different from histone methylation, and addition of this methyl groups to DNA also represses transcription. Usually the cytosine, one corner of the cytosine, a methyl group is usually added by some enzymes, and that cytosine molecule, cytosine nucleotide is usually next to a guanine. So this is how the structure looks like. This structure, when you have CG repeats with one of the C methylated, it's referred to as a CPG island. They call it the CPG island. CPG, where P represents the phosphate group in the DNA backbone. So when you say CPG islands, you are referring to the DNA directly where the cytosine nucleotide is linked to a methyl group, which means that gene expression is repressed. Okay? But you don't use the word suppressed, you use the word repressed. So it's repressed. There is also other level of gene expression. Here we are looking at multiple transcription factors, recognizing multiple consensus sequences. And if you look at it, see, this is your DNA, right? And this is your promoter. This is a regulatory promoter. Basically, a regulatory promoter or promoter is still the same region, but depending on what kind of transcription factors sit here, what other kind of transcription factors sit here, you call it a core promoter, you call it a regulatory promoter, you call it what is that for lipoprons? I forgot in a second. So lipopron also, the regulatory protein binds what? The regulatory protein binds a part of the promoter gene. What is it called? I forgot it. Operator. Thank you. Operator. So operator is also part of the promoter. So see, these two slides are all about, this is a piece of DNA, it has multiple consensus sequences, so multiple proteins bind on it. These multiple proteins can interact with one another and lead to gene expression. Multiple proteins binding will also enable the RNA polymerase, which is also a protein, to sit very comfortably on it and slide over it. You get that point? So this is all about multiple consensus sequences in the promoter region, multiple transcription factors, and RNA polymerase leading to gene expression. So if this is the promoter, there's also a piece of the DNA referred to as an enhancer, which is usually a little far away. Once again, enhancer is also a DNA, but it also has consensus sequences. Now, there are some other special transcription factors which bind to the enhancer in such a way that they bend the DNA so that this transcription factor here, the one in purple, is actually interacting with the other transcription factor, and it can influence the rate of transcription. Get that? So here if you see, there is this transcription activator protein, which is probably one of the transcription factors sitting here. There is one other transcription factor, it's referred to as the mediator. This transcription factor here in light purple interacts with this dark purple transcription factor through another protein called coactivator. So basically you have multiple transcription factors and cofactors which interact with one another by sitting on consensus sequences, by recognizing them, enabling the RNA polymerase also to come and sit comfortably and read the gene. Is that clear, guys? So look at this. General transcription factors are part of the basal transcription. So here you see this basal transcription apparatus. Do you see that? So here in this basal transcription apparatus, what you have is this long yellow one is the RNA polymerase. This one which I am pointing with the mouse. Now it has recognized the Tata box. Okay? There are, you see some of the small circles or elongated elliptical circles. These are some transcription factors. In fact, RNA polymerase with some of these transcription factors here can lead to some amount of transcription. Basically, you can have some molecules of RNA synthesized, but if you want to have more transcription, basically high amount of RNA synthesis to happen faster and all that, then you need this special transcription factor here. You also need this special transcription factor sitting on the enhancer to bend over and interact with this complex and then influence or increase or usually increase the rate of transcription. Get that? So in other words, if you don't have your specific transcription factors with some basal apparatus, which is some basic transcription factors and RNA polymerase, you can have some amount of transcription happening. But to have an enhanced level, to have higher transcripts, you need other transcription factors sitting on the enhancer, other transcription factors sitting on the regulatory promoter instead of the, in addition to the core promoter to allow transcription to happen. So activator proteins bind the consensus sequences and interact with other components of the basal transcription apparatus and influence the rate of transcription. Okay, activator proteins. Okay, right, okay. So some of the proteins are referred to as mediators because they communicate with other transcription factors to some other proteins called co-activators. Once again, like bunch of proteins recognizing a bunch of consensus sequences, interacting with one another, leading to transcription. So all these are also examples. But now, for this example, which I just said, bunch of proteins and a bunch of consensus sequences, here we're going to go through an example for GAL4. So regulation of galactose metabolism through GAL4. So galactose is a sugar which needs to be metabolized, which means it needs to be broken down and used up by the system. So if galactose needs to be broken down, obviously you need some gene expression, some proteins have to be synthesized which can metabolize galactose. Now here, one activator protein called GAL4 in yeast is actually required for galactose metabolism. GAL4 gene in yeast. Now, just like the lac-operon genes that control galactose metabolism, which are inducible, likewise here GAL4 can also be induced. So when galactose is absent, these genes are not transcribed. When galactose is present, genes are transcribed. This is actually for the lac-operon which we studied. So we studied that galactose comes and binds to the regulatory protein in such a way that now the regulatory protein cannot bind on the promoter region, leading to. So likewise, here we'll see how this GAL4 can kind of regulate galactose metabolism. So one way that it works is, sorry, it's confusing. So GAL4, which is actually a regulatory protein, right? I said here, it binds a protein called GAL8, which regulates GAL4 activity. So binding of the GAL4 to another protein called GAL8 will regulate the GAL4 activity. How does it do that? When galactose is not present, so basically galactose here is a substrate which needs to be metabolized. But here galactose can also have the ability to bind some of the proteins and regulate expression. So what does galactose do? When galactose is absent, if you don't have galactose, this GAL8 will bind the GAL4 and prevent GAL4 from activating transcription. So normally here GAL4 is a transcriptional activator protein. GAL4 is a regulatory protein which is a transcriptional activator. Now when you don't have galactose, GAL8 binds GAL4 and it prevents the GAL4 from activating the structural genes here, so to speak. When you have galactose, let's say if you have galactose binds another gene called GAL3. This galactose plus GAL3, they will interact with GAL80 in such a way that the GAL80 cannot stop GAL4 from doing its job. So this is an example where you have multiple proteins interacting with one another to either positively regulate expression or repress expression. So let's look at that in the form of an image. I think it's probably the best way to remember. Look at this here. So you have GAL4. which is a regulatory protein shown here in violet. It binds the DNA, it wants to promote gene expression, but then GAL80 is bound to GAL4 as well, and it represses transcription. And all this is for regulation, for the expression of genes which can metabolize galactose. But here galactose is also, it's not only the gene that, it's not only the sugar that needs to be broken down by these structural genes, but here when you have galactose, what it does is it binds GAL3. Now this GAL3 plus galactose, which is a complex, these two will displace this GAL80 from GAL4. So here you see you have GAL4, but you don't have this GAL80 here. So when there is no GAL80, this GAL4 now interacts with other transcription factors which are linked to the RNA polymerase, leading to transcription. So here, galactose is not only the metabolite where these structural genes will act on, but galactose is also able to regulate the expression of its structural genes by binding to some other proteins like GAL3, which will displace this GAL80 from binding to GAL4. Is that clear, guys? So, transcriptional repressors. These are also, transcriptional repressors also, regulate gene expression, but obviously when they compete for the binding site where this transcriptional activator proteins will bind the promoter. So when transcriptional activator proteins bind the promoter, when I say transcriptional activator proteins, I'm referring to transcription factors once again. So remember, transcription factors need not promote a gene expression all the time. They can be activators, they can also be repressors. Here, some of the inhibitory proteins, which are basically repressors, they can compete with some of the other transcriptionally activating proteins for the binding sites and lead to inhibition or lead to repression of gene expression. Now, there are other regions on the DNA referred to as enhancers and insulators. I think enhancers is what we actually covered like a couple of minutes ago. These enhancers usually, which I spoke earlier, they're usually much far away from the gene that they activate, but they can be very close to the gene because the DNA can bend over. That's how transcription factors binding to enhancers come in direct contact with RNA polymerases or other proteins for transcription. Now, insulators. Insulators are also regions in the gene which also have some consensus sequences, and some other special insulator binding proteins will bind on these insulator regions which regulate gene expression. So we have spoken about enhancers, what is that? Promoter. We spoke about, and we said core promoter. We spoke about promoter regulatory. Regulatory promoter. Then now we spoke about enhancer. We now speak about insulators. Remember, all these are actually some consensus sequence of the DNA, to which all these transcription factors bind. All these are proteins that bind the DNA at consensus sequences. So here now we are listening to the fourth new term called insulators. Now insulators are DNA sequences that block the effect of enhancers in a position dependent manner. If the insulator lies between an enhancer and a promoter, it blocks the action of the insulator. So let's look at it. So see this. This is gene A here. We have gene B here. This is the promoter for gene B. This is the promoter for gene A. So transcription is happening this way, transcription happening this way. Now, enhancers here, this enhancer is a region of the DNA where some special transcription factors can sit and increase the rate of transcription. Likewise, another transcription factor can sit here and increase the rate of transcription from this gene. But if transcription factor sitting on enhancer 1, if they want to increase the expression of gene B, they cannot do it because this region called the insulator, where some other insulator binding proteins are present, they prevent this enhancer 1 in positively promoting the expression of gene B. Likewise, if enhancer 2 wants to positively promote the expression of gene A, this insulator prevents it because the insulator lies between these two genes. So this enhancer can positively promote the expression of gene A, and enhancer 2 can positively promote the expression of gene B, but enhancer 2 cannot positively regulate the expression of gene A because the insulator proteins bound to the insulator region are actually lying in between. You get that, guys?
So if that wasn't there, enhancer 1 can promote gene B?
Sorry?
So if the insulator wasn't there, the enhancer 1 could promote or stimulate gene B?
Yes.
And vice versa?
In some cases, yes, yeah. So, I mean, yeah. So, this is just to say that those enhancers, but if they lie between the enhancer and the promoter, they kind of prevent the positive regulation of enhancers. So like this slide reads here, insulators are DNA sequences that block the effect of enhancers in a position dependent manner. I wrote it position dependent because they kind of lie in between. If an insulator lies between an enhancer and a promoter, it blocks the action of the enhancer. So gene expression can also be regulated in a way which depends on the let's say on external heat or based on temperatures. For example, here we speak of an example of gene expression as to how regulation of transcriptional stalling and elongation happens. Stalling is basically a part of the a protein is synthesized and then the ribosome stops there. So this is also a different mechanism of gene regulation. So you see that in prokaryotes we spoke about operons, and we spoke about regulatory proteins and structural genes and a few more, and we stop. But here, if you're looking at eukaryotes, we speak about DNA methylation, histone methylation, acetylation. We speak about now we speak about heat shock protein. We speak about multiple transcription factors binding to one another. We speak about insulators. We speak about insulator binding proteins. We speak about enhancers. So all this that we speak about is to show how many levels of gene expression can happen in eukaryotes. So here is one more level where we show how protein synthesis is actually stopped in between because of some proteins called negative elongation factors. Now here, what we're trying to say is there are some proteins called heat shock proteins. What does this heat shock proteins do? Normally, if an organism is being exposed to very high temperatures, for example in Drosophila, beyond temperature, Drosophila cannot live. I mean, like optimally they're put between 18 degrees centigrade and 23, 25 degrees centigrade. But let's say beyond 30 or into 35, they usually die. So let's say a Drosophila is exposed to 30 degrees centigrade, which is not very comfortable for it. So in order for the system to be functional or for the fly to be kind of doing good, proteins called heat shock proteins are synthesized. In fact, if the fly is in ambient temperatures where it's comfortable, heat shock proteins are not completely synthesized, but the ribosome slides over this heat shock gene, heat shock mRNA, produces part of the protein and it stops there. Only when the fly is exposed to higher temperatures, these negative elongation factors are removed so that the remaining part of the heat shock protein is synthesized very fast. This is also a different level of regulation of gene expression at the ribosomal level. So the ribosome is bound to the messenger RNA. It synthesizes a few amino acids, and then proteins called negative elongation factors play a role here. Only when the temperature is kind of ambient, or basically only when the temperature is high, the remaining part of the heat shock protein is synthesized so that the fly can withstand higher temperatures. Okay? So this is gene regulation at the ribosomal level. Now, gene regulation also happens through RNA splicing. So we have looked at splicing. We know that RNA has introns and exons. Introns needs to be removed, and if introns have to be removed, there are some certain sites within the intron referred to as certain consensus sequences or some regions where spliceosome machinery recognizes the introns and removes them off. So splicing, depending on how splicing happens, so if splicing is here, one protein is synthesized. If splicing is a different spot, a different protein is synthesized, and that different protein also has a different job. So gene regulation can also happen at the level of splicing in eukaryotes. Here, they give you an example of something called a T antigen gene, which is found in SV40 virus. So this gene called T antigen will actually enable the SV40 virus to replicate very nicely in host cells, depending on the surroundings or the situation. The T antigen, the small T antigen or the large T antigen gene is actually synthesized. Now, how does the small T antigen gene or the large T antigen gene be synthesized? That once again depends on splicing mechanism. So depending on the situation or the surroundings, splicing can happen here or there, leading to a smaller T antigen protein or a larger T antigen protein, and both these proteins are required by the virus to grow or to grow in the host cells, and splicing for that gene or the T antigen gene depends on depending on the surroundings, which actually these T antigens will allow the virus to replicate in host cells. And so splicing depends on the surroundings and the situation. So this is also a mechanism to show that gene regulation happens also at the level of differential splicing. Remember, this is gene A, this is gene B, this is an intron. Normally introns are supposed to be removed, but also so splicing should happen here, mentioned with number 2, but also splicing can happen here within exon 1 and exon 2, which is represented as A and B, and that would lead to the synthesis of this protein or a different protein. So splicing also regulates gene expression in eukaryotes. Now, alternative splicing so for example, for that, the code here is splicing in Drosophila. I think we have looked at the information on this slide was actually taken from chapter 6, I suppose, because in Drosophila we studied that the presence of two X chromosomes actually determines female gender. Presence of only one X chromosome determines the male gender. So less X chromosomes favors males, more X chromosomes prefers females. We said that, I mean, once again, this is an example quoted to show how splicing actually regulates gene expression in eukaryotes like Drosophila. Here, if you have two X chromosomes, a female specific promoter, basically a gene, one specific gene is actually activated in females, which leads to the synthesis of a gene called sex-lethal gene. This gene, the protein of this gene, is involved in splicing of another gene called transformer. And this transformer gene, along with its second partner called transformer 2, they together regulate the expression of a gene called double-sex genes. These double-sex gene promotes the female characteristics. Now, what happens if you don't have two X chromosomes? So, if you have two X chromosomes, sex-lethal gene is activated, right? If you have only one X chromosome, what happens is the splicing of this transformer gene is different. So sex-lethal gene, the splicing is actually different, which leads to a non-functional transformer gene in males, which actually favors the male phenotype. Let's look at that slide here. So look at this. So this is sex-lethal gene, SXL gene, right? It's activated in females which have two X chromosomes. Now the sex-lethal gene will splice the messenger RNA for transformer genes. So transformer gene 1 and transformer gene 2, they both regulate the expression of DSX, which is double sex gene, which favors a female phenotype. Now let's say here you don't have two X chromosomes, you only have one X chromosome. Now sex lethal gene, there is no sex lethal protein synthesized. If the sex lethal protein is not synthesized, the splicing of this transformer gene is different. So when you have sex lethal protein, transformer gene is spliced in a different way. In the absence of sex lethal gene, transformer gene is actually spliced in a different way, which leads to a non-functional transformer gene. That non-functional transformer gene cannot act on the double sex gene, which eventually favors the male phenotype. You see that? So here, this is a very good example to show you that expression of a certain protein leads to certain kind of splicing that eventually down the stream leads to female. Now, in males, if that protein is not expressed, splicing is now entirely different, which down the stream promotes the male phenotype. Is that clear? Yes, splicing. Okay. Also, the other level of gene regulation in eukaryotes is actually at the RNA level. We have studied about small interfering RNAs and microRNAs. I told you that the tips of some messenger RNAs, when they fold on themselves, the enzyme called dicer recognizes this double-stranded DNA, it cleaves it into pieces. These small pieces, which are the tips of messenger RNA, will fall off. These small pieces now start having some regulatory function. They are now referred to as small interfering RNAs, small microRNAs. They can go and bind to other messenger RNAs, creating double strands, where some other enzymes recognizes double-stranded RNA molecules and cleaves it into pieces. So enzyme dicer cleaves the process double-stranded RNA to produce single-stranded small interfering RNA and microRNAs. They combine these small interfering RNA and microRNAs, they combine with some other proteins and they induce something called RNA-inducing silencing complex, or also referred to as a RISC. This RISC will either inhibit translation or destroy the messenger RNA, thereby there is no translation. I mean, I'm skipping this, I'm going a little fast on this because these are something that we have dealt exclusively in the previous chapters. So, RNA can actually, the microRNAs can actually cleave the messenger RNA, they can, or they degrade it completely, they can bring about transcription silencing, or they can inhibit translation. So these microRNAs and small interfering RNA can do not only inhibition but also destroying the messenger RNA for other genes. I will show. Okay, look at this here. Double-strand RNA, you have SI, it's broken into small pieces by dicer. This small piece of RNA combines with this, which is another enzyme complex. They go bind to some other messenger RNAs and cleave the messenger RNA and destroy it. Or, in this case, if it's a microRNA, what it does, it binds to the RISC, it binds to some other messenger RNA. It does not destroy the messenger RNA here in this case, but it actually inhibits translation. So either way, they do not promote, they kind of regulate protein synthesis in a negative way. Okay? Okay. Okay. I'm just looking at the time that we have for another chat. Now, transcription silencing. There is also other complexes of RNA with proteins referred as RISC, which is RNA-induced transcription silencing, which is analogous to RISC complexes. They also regulate gene expression by binding to other messenger RNAs and preventing translation from happening. Silencer-induced RNA degradation, which we have looked up here in this slide. We spoke about RISC, where siRNAs bind and cleave the messenger RNA here. Then here miRNAs bind to the messenger RNA. They don't cleave it because there is no exact complementarity. You see this bump here, which means there is no exact complementarity between the messenger RNA and the microRNA. In this case, it does not destroy the messenger RNA, but prevents translation from happening. Okay. So also, you also see gene regulation because of some proteins called initiation factors. Here they give an example of T cells. If T cells are activated because of some viral infection, you need to have a lot of protein synthesis happening. In this case, some proteins called initiation factor called initiation factor T, initiation factor 3. It enables the ribosomes to actually synthesize more and more copies of protein from one messenger RNA. Here you see that T cell is infected with some virus. The T cell now recognizes the virus. It activates. Now these initiation factors, what they do is they go bind this ribosome, these red spots here. They kind of enable the protein synthesis to happen faster and faster. So that, this is also a level of regulation where protein synthesis, the synthesis of proteins are actually increased 5 or 10-fold because of special proteins called initiation factors. So all these 10, 12 levels which we discussed till now are different levels at which gene expression is regulated in eukaryotes. So obviously you know that gene expression in eukaryotes is much, much, much complex compared to prokaryotes, which operate through the operon concept, prokaryotes, right? We'll take a 10-minute break and we'll go to chapter 18.